Now, as I mentioned before, genetics is often blamed for Alzheimer's disease, especially the well-known APOE4 gene. The APOE gene can be found on the 19th chromosome, and it comes in 3 flavours: APOE2, E3 and E4, and everyone has 2 of these genes, one from each parent, and it is the combination of 2 APOE4s which is most strongly associated with Alzheimer's disease. And having 2 of these genes confers probably about a 5 times greater risk of developing Alzheimer's disease. But here's the thing, genetics is not fate. Just because you happen to have 2 E4 genes does not consign you to a fate of dementia. It is, I believe, dependent on metabolic health. Those in good metabolic health, irrespective of E4 status, do not have an increased risk of Alzheimer's disease. The APOE gene serves as the blueprint for this protein, which is embedded within the membrane of HDL cholesterol particles. And HDL particles in the brain act as a natural defense against beta amyloid deposition, because it can actually remove them. That's right, HDL particles in the brain, by way of the APOE protein, can remove beta amyloid. Now, the APOE3 variant is by far the most common, while the less common APOE4 is the one that gets all the bad press. This is the one that is especially vulnerable to damage because of its unique molecular structure. You'll recall how beta amyloid peptides can be damaged both by sugar stress or glycation and oxidative stress from oxidized oil consumption. Well, the APOE protein can also be damaged in the same ways. And it just so happens that the E4 variant, by virtue of its particular molecular structure, is especially prone to this. We have clear evidence from this paper which compares the tendency of the APOE3 protein to form advanced glycated end products to the APOE4 protein. The E4 variant is damaged by advanced glycated end products at 3 times the rate. Now consider for a moment what this means. If you carry APOE4 and are not metabolically healthy, the HDL particles in your brain which remove toxic protein deposits are less likely to be able to function. This tendency for glycation damage, if you were not metabolically healthy, is why those with the APOE4 genotype on average are about 5 times more likely to develop Alzheimer's disease. And when you consider that about 98% of US adults over the age of 60 are not completely metabolically healthy, you can see why having the APOE4 gene might not be such a good thing. The thing is, if you are metabolically healthy into old age, then having the APOE4 variant likely doesn't matter. The formation of advanced glycated end products is a manifestation of metabolic disease. And there's also other consequences of dysfunctional HDL particles which occur due to glycation and oxidation stress. This study looked at the activity of an enzyme called matrix metalloproteinase 9 in HDL particles, which were normal, and those which were dysfunctional. And it was clearly shown that when HDL particles became dysfunctional, the activity of this matrix metalloproteinase 9 increased dramatically. And the reason this is a problem is because matrix metalloproteinase 9 has been shown to disrupt something called the blood-brain barrier. The blood-brain barrier basically prevents certain circulating substances in our blood from crossing into the nervous system, where our brain resides. And disruption of the blood-brain barrier is an early biomarker of Alzheimer's disease. And this significance was demonstrated in this paper from last year, which was published in Nature. It clearly showed increases in blood-brain barrier dysfunction, which were caused by matrix metalloproteinase 9 levels, were higher in those carrying the APOE4 protein, making their HDL particles prone to damage. So the link is, carry APOE4, your HDL is more likely to be damaged. That reduces your ability to remove the beta amyloid plaques and leads to disruption of your blood-brain barrier.